Okay, thank you. First, I want to thank the organizing committee for this kind invitation. So today I would like to convince you that high-throughput sequencing is revolutionizing the field of infertility by providing for the first time a molecular etiology for most of the patients suffering of severe infertility. So I have no conflict of interest. So just I would like to start by a few slides concerning infertility. So infertility is in fact more common than most of the people think because around 15% of couples fail to obtain a baby after 1 year of unprotected intercourse. Female and male are equally affected, and we found also a female and a male factor in 1/3 of couples, and usually corresponding to association of subfertility factors. So there is a lot of causes for human infertility, including genetic defect, environmental factors such as endocrine disruptors, also congenital or acquired defect. And most of these factors, in fact, lead to altered gametogenesis, preventing in vivo fertilization. Then you have two cases. First, if you have oocyte of good quality, then you can use abnormal sperm for in vitro fertilization using assisted reproduction technologies. For most of the couple, the technology is successful and then the couple get the babies. However, for some couples, the technology fails and the couple remains childless. So in the other case, when you have no sperm, a condition known as azoospermia, or also if the oocyte is of bad quality, then you cannot use assisted reproduction technologies, and then again, the couple remains childless. For all these couples remaining childless, clearly new therapeutic strategies are necessary. So the discovery of new therapeutic strategy involves a better understanding of the molecular etiology of infertility, and we think that high-throughput sequencing offers now a great opportunity to discover the main causes of genetic infertility. But also, we think that a better understanding of gametogenesis is necessary, and it is the reason we do a deep study on the function of the protein characterized on the step 1. So here is our scientific flow chart. So we start to discover candidate genes, and then a very important step for us is to validate these candidate genes through the generation of animal models. Using these animal models, we can get a better understanding of the molecular pathogeny, but also on the function of the protein and therefore the gametogenesis. And finally, with all these results, you can develop new therapeutic strategy. So concerning the discovery of candidate gene, we start from cohort of infertile patients with no other associated pathology, which is a very important point, coming from different countries. So we have a collaboration in North Africa, in Iran, and also a lot of patients coming from France. So with the DNA of the patient, we do exomic sequencing, so we decided to work on the exome because we know that 85% of pathogenic mutations are exomic. And then using homemade algorithm, we do exomic sequence analysis, which allow us to identify new candidate gene. So next, we want to go further, and so it is the reason we generate or obtain animal models. So you have two ways to get it. So now you have around 6,000 knockout mice already done in the repository collection, and you can work either with the European IMPC or the American collection. You can also generate your own knockout or knock-in mice or Cas9 system, and we have a long experience doing that in my labs, and it's very easy. Then you can get your knockout in less than 8 months now, so it's quite fast. And then with all animal models, you can study the impact of the absence of the protein on spermatogenesis and on genesis, and finally to identify the function of the protein during gametogenesis. So my talk will be divided in 2 parts. The first one concerns male infertility and the second one female infertility. So male infertility is a very polymorphous, and you can have a lot of type of defects such as a morphological defect, a condition known as teratosospermia affecting either the head or the flagellum. You can also have quantitative defect affecting the number of sperm. So if you have low count sperm, a condition on oligosospermia or no sperm azoospermia. And finally, you can also have functional defect, such as a sperm unable to move or also sperm unable to activate oocyte. So in my lab, we have already characterized 80 genes involved in these different conditions. So why it doesn't? Okay, so we have found most of our genes were characterized in teratosospermia, but also we have characterized 1 gene in azoospermia and 1 gene in activation defect. I have no time to talk to you about all these genes, so today I'm going to talk only about 1 gene involved in azoospermia, which is a SPINK2. So we started in this study from a French consanguineous family presenting 2 infertile brothers. So spermatocytogram clearly show that 1 brother was completely infertile with no sperm in the ejaculate, whereas the second brother was severely oligosospermic with a very few sperm in their ejaculate. So we studied the structure of the seminiferous tubule by histology, and you see clearly that in the patient, you have spermatogenic cells, so clearly indicating that, in fact, you have a block of the spermatogenesis, and also you have a deep disorganization of the structure of the seminiferous tubule. So from the DNA of the 2 brothers and the parents, we did DNA exomic sequencing, and we focused in particular on homozygous mutation because we think that this disorder is likely recessive. So we found, in fact, 6 genes presenting homozygous mutation with damaging prediction. And 1 was very interesting because it was highly and predominantly expressed in the testes. This gene is a SPINK2, and SPINK2 in fact codes for protease inhibitors belonging to the family of the serine peptidase inhibitor Kazal type 2. So the gene is made of 4 exons, and exon 3 and exon 4 code for the inhibitory site of the protein. So we found a splicing mutation just before exon 2, leading to the production of 2 transcripts, in fact leading exon 3 and exon 4, and therefore producing non-functional protease inhibitors. So we obtained the knockout from the reproductive collection, and we studied the reproductive phenotype of the male. So first, as you can see, when you cross the male with the wild-type female, no pups were obtained, clearly showing that the male are completely infertile. And also, when you do a spermatocytogram of cells collected in the coda epididymis in mice, you see clearly that you have no sperms, so clearly indicating that a SPINK2 knockout mice mimics the human condition with a strong infertility and a complete azoospermia. So to go further to study this azoospermia, we studied the structure of the seminiferous tubule, either by histology or by immunofluorescence. So, and we observe clearly that in the knockout, you don't have any condensed or elongated spermatid. Moreover, as you can see here in the round spermatid of the wild type, you have a purple dot corresponding to the acrosome. And clearly here in the round spermatid of the knockout animal, you have no purple dots, clearly suggesting that the biogenesis of acrosome is defective. So to confirm this point, we use a marker of the acrosome with an antibody, again a protein known as SP56. So you can see here the nice staining corresponding to the acrosome, and you see clearly that you have any staining, no staining in the section of the seminiferous tubule of knockout mice. So clearly, acrosome is absent in the knockout spermatid. So next we wanted to see the localization, so using non-made specific antibodies. So again, you see here clearly, so the green staining that the antibodies localize the protein in the acrosome, so which is validated with the co-staining with the marker of the acrosome, SP56, and this nice co-localization. The antibody is specific because you don't have any staining on the knockout. And in human, you have exactly the same staining over the acrosome. And also, it's worth to note that the staining remains in the acrosome in the mature sperm in both rodent and human. So, acrosome is made of the fusion of many vesicles coming from the Golgi apparatus. So, we wanted to know what is the impact of the absence of SPINK2 on the Golgi apparatus. So to do that, we do a double staining with one antibody staining the Golgi apparatus, GM130, and also one antibody staining the nuclear envelope facing the acrosome, here the dumpy19L2. And we use these antibodies because you have no acrosome on the knockout, so you cannot use SP56. So you can see here in the control one spermatid that the Golgi apparatus is packed and facing the acrosome. So in contrast, when you look at the localization of the Golgi apparatus in knockout round spermatid, you see clearly this very odd localization. Moreover, you see that you have different red dots corresponding to the Golgi apparatus, clearly indicating that the Golgi apparatus is fragmented. Therefore, the lack of SPINK2 induces a destabilization of the Golgi apparatus. So here I have to do a recall concerning the role of protease in sperm. So proteases are very important in sperm physiology because when sperm arrive close to the cumulus cell, surrounding the oocyte, then you have to cross these layers. So there is an exocytotic event known as acrosome reaction, and this acrosome reaction releases different enzymes, among them proteases, which are necessary for sperm penetration through cumulus and zona pellucida and allows the sperm to reach the ooplasma. Among the different proteases, one is very important, it is aquasine. So aquasine is a trypsin-like protease. So aquasine is synthesized as a proaquasine, and if the pH is above 6, then you can have an autoactivation of the proaquasine, and then you can get an active acrosine. So we made the hypothesis that SPINK2, in fact, is able to block the autoactivation of proaquasine and then allows normal cell proliferation. In contrast, in the patient, in the absence of SPINK2, you have autoactivation of acrosine, and then you have autolysis and cell death. So to confirm this hypothesis, we followed the cell proliferation, the hex cell proliferation. So here in the red purple, the curve, the purple curve, you see it corresponds to the control proliferation of hex cell. And then we decided to transfect a plasmid containing the aquasine. And you see clearly here in the red condition that the aquasine stops cell proliferation and even induces cell death. However, if you co-transfect in hex cell a plasmid containing aquasine and SPINK2, here it is a blue curve, then you can see clearly that you can rescue the normal cell proliferation. So clearly indicating that SPINK2 is able to prevent autoaquasine activation. So This result allowed us to propose molecular pathogeny of SPINK2 azoospermia. So in the proacosine, acosine is synthesized in the reticulum, transit through the Golgi apparatus before reaching its final location, which is the acosome. And we know that the pH of the reticulum and the Golgi apparatus is above 6, therefore clearly allowing the autoactivation of SPINK2. So to prevent this autoactivation, you have a co-synthesis of proacosine and SPINK2 within the reticulum, preventing autoactivation and then normal spermatogenesis. In the patient, you have autoactivation, and then the active acrosine is able to destabilize a Golgi apparatus, then preventing acrosome formation and release of the protease within the cytoplasm, and then cell death and azoospermia. So, is the SPINK2 mutation a common cause of spermatogenesis failure? So to address this question, we sequenced the gene SPINK2 in 611 patients presenting either azo or oligozoospermia. And then we found only one patient with an heterozygous mutation, which abrogates the SPINK2 start codon. To validate the pathogenicity of this mutation, so we transfected HEK cell with either a plasmid containing a wild-type SPINK2 or a plasmid containing the mutant SPINK2, and we followed the production of the protein by Western blot. And so you see clearly that for the wild type, you have a nice signal corresponding to the production of SPINK2, and in the mutant, you have absolutely no signal, so clearly indicating that the mutation is pathogenic. So when you look at the spermatocytogram of this patient, you see clearly that is oligozoospermic. It's what it is expected because it comes from an oligozoospermic cohort, but also he has a strong motility defect. So this patient is oligozoospermic but also present as stenozoospermia. So when we did the phenotype of the male in the mouse, so we didn't check, in fact, carefully the reproductive phenotype of the heterozygous mice because we were thinking that it was a recessive disorder. However, from the human result, we decided to readdress this question. So we didn't see any defect on the number of pups produced by the heterozygous male, but we see different defect. So first, an increase of the abnormal form, and in particular in the head. But also the mice display low motility, so clearly indicating that the absence, so the decrease of the SPINK2 concentration within the cytoplasm, in fact, has a defective impact during spermatogenesis. So this, from human result and mouse result, we can say that SPINK2 haploinsufficiency leads to oligozoospermia, whereas SPINK2 absence leads to azoospermia. And we think that, in fact, oligozoospermia and azoospermia can be seen as a continuum. So now I am going to talk about my second part, which is a gene involved in female infertility. And in particular, we're going to talk a gene which is very important to meiotic maturation. So among the different female infertility, there is one known as oocyte maturation deficiency, and which is very poorly explored. It is a rare form of primary female infertility, which consists to the repetitive production of immature oocyte of female after hormonal stimulation. So usually, after oocyte growth, you have induced by a surge of LH, meiotic maturation, leading to the production of mature oocyte. In this patient, in fact, oocyte are arrested at different stages, either the GV, the M1, but also the M2 stages. And the quality of the oocyte are very bad because these oocytes are immature, then in vitro fertilization is not possible. This condition is clearly distinct from primary ovary insufficiency because menstrual cycles are maintained, And there is only one cause, genetic cause already identified for this syndrome. It is a mutation in a specific human tubulin gene, which is TUB8. So we applied exactly the same phenotype for this patient. So we started from a cohort of infertile female, and so we started for 33 women from North African origin, so most of them coming from Tunisia. And we did whole exome sequencing on this patient, which allow us to identify a list of gene candidates. And then again, we decided to validate our study using animal models and knock-in mouse model. So this strategy allowed us to identify a stop mutation in exon 6, leading to the formation of a truncated protein missing a very important functional domain in the protein, which is a PAT1 domain, and then therefore leading to the production of a non-functional PATEL2 protein. So interesting, so this mutation was found in 26% of the patient, and interestingly, all the patients have exactly the same mutation. So we decided to compare the infertility parameters of the female of our cohorts. So we divided this our cohort in two groups. So the female with presenting a PATEL2 mutation, here's the gray column, with the female presenting no PATEL2 mutation, the white column, with a large cohort of a patient having undergone hormonal stimulation in the frame shift of a male factor infertility. So here corresponding to the blue column. And then you can see that when you look at the total number of oocytes retrieved, there is no difference between the different groups. So now if you look at the oocyte produced during this hormonal stimulation, so in blue, so the control cohort, you see clearly as expected that most of the oocytes are in the M2 stage, which is the mature oocyte stage. However, for the PATEL2 mutated patient, most of the oocytes are in the GV stage. So it is important to know that for the TUB8 cohort, most of the oocytes were blocked in the M1 stage, so clearly indicating that the PATEL2 mutation, in fact, is an interesting mutation leading to an original condition. So what is already known about PATEL2 protein? So when we started this work, in fact, nothing was known about mammalian PATEL2. However, there is one autolog in Xenopus, because there is no autolog only in vertebrates, so only vertebrate you can see PATEL2. And so in Xenopus, PATEL2 is an oocyte-specific RNA binding factors which control the translational repression of maternal mRNA, and it forms a part of a large ribonuclein complex, including CPEB and MSY2. So, and in human, from the expression database, you see that PATEL2 is highly expressed in the oocyte with no expression in the cumulus cell. And when you take both the Xenopus data and the human data expression, then clearly indicating that PATEL2 is a very good candidate to explain the OMD syndrome. So again, we decided to validate this mutation by generating a PATEL2 knockout mice. So we wanted to reproduce similar phenotypes, a similar protein that is a protein missing the PAT1 domain in the mouse. So for that, what we did, we excised exon 7 and we realized a frame shift just after exon 7 downstream, therefore allowing to produce a non-functional protein. And we use this knockout lineage to address two questions. Is PATEL2 necessary for normal oocyte maturation in mice? But also, what role does PATEL2 play in oocyte maturation in mammals? So first, we started by a reproductive phenotype of the female. So we cross here in blue wild-type female with wild-type male, and in red knockout female with wild-type male. And you see clearly that over a period of 6 months, so we're able to measure the number of liters, but also the number of pups per liter. And so you see clearly the difference. So the knockout has less liter per over the period of 6 months, but also less pups per liter. So clearly indicating that the phenotype is a little bit different because the female were completely infertile, but in the mouse, it's a severe subfertility. So what was the morphology of the oocyte retrieved after hormonal stimulation? So first, when you count the oocyte collected after superovulation between knockout female and wild-type female, you see there is no difference, and it was expected because we didn't see a difference in the human. However, we were very surprised because in contrast to human, we obtained oocyte presenting a frost-polar body. So clearly it is a M2 oocyte, a mature oocyte. So it was a big difference between mouse and human. However, when you count the number of M2 oocyte, you see clearly that there are higher proportion of a non-M2 oocyte in the oocyte collected, so clearly indicating that there is a defective meiotic maturation leading to a decrease of M2 oocyte. So what is the morphology of this M2 oocyte? So we focused in particular in two aspects. First, the size and the shape of the spindle, using an antibodies against tubulin here in red, but also the localization of the chromosome here in blue with the X marker. And so here is the wild type, that is a normal configuration. And in the knockout, you see clearly that the M2 oocyte have several defects, such as abnormal spindle, or also you can see here an abnormal spindle associated with a misaligned chromosome. And also here you see the spindle is rotated, so instead to be parallel to the ooplasma, it's perpendicular. So in fact we see a lot of defect in this M2 oocyte, abnormal spindle, misaligned chromosome, spindle rotation, but also numerous aster. You can see here all these aster of tubulin, and clearly indicating that not only the number of M2 oocyte is lower, but also the M2 oocyte produced are of bad quality. So we wanted to know how the M2 oocyte can sustain the fertilization. So we use oocyte coming from superovulation, and we did in vitro fertilization. So in the wild-type condition, you can see here these nice 2 pronuclei 9 hours after fertilization. And in the M2 oocyte, from the knockout mice, you see there is a lot of defect. Either you have only one pronucleus, or you have also partial decondensation of sperm nucleus, or here in this case associated with polyspermy. So we see a lot of different defects in the zygote obtained with knockout mice. And here you see all these defects are presented here in these charts, and partial decondensation, not 2 pronuclei, polyspermy, and mixed defect. And again, what does it mean? It means that the maternal factor necessary to activate fertilization are not present. So we want you also to know the developmental competence on this oocyte. So to do that, we did a little bit a different protocol because we think that superovulation can modify the developmental competence of oocyte. So we used non-stimulated female, and we made in vivo fertilization. Then we collected oocyte 12 hours after in vivo fertilization, and we cultured this embryo for 4 days up to the blastocyst state in vitro. Then we can follow the progression of the embryo development. And also at the end, we are able to compare the number of blastocysts with the number of pups obtained with full in vivo experiment. So you see clearly that when you collect in the wild type, so when you use oocyte coming from the wild type cross with a wild type male, you see clearly is that all the zygote collected develop perfectly up to the blastocyst stage. And moreover, you see that the mean number of blastocysts is similar to the mean number of pups, and so clearly indicating that there is a very good developmental competence in wild type. The situation is clearly different when you use oocyte coming from a knockout female. So we collected a little bit more, we don't know why in this situation, oocyte, and you see clearly that we have abortion or a stop of development in all the different stages observed. And moreover, you can see that there is a strong difference between the number of obtained blastocysts and the number of pups. So clearly suggesting that maybe the blastocysts are of poor quality. So to validate this hypothesis, we in fact measured the morphological aspect of blastocyst. So in the control, so when you use wild type oocyte, you obtain blastocyst of a very good quality. So here in this one, you have a blastocyst hatching. And when you use knockout oocyte, you see clearly that you have a different type of defect affecting blastocysts, such as a higher number of blastocell, but also several inner mass cell masses. So clearly indicating that the embryo development is impaired. So if we want to summarize, so the Patel2 mouse oocyte meiotic deficiency, so we have shown that there is a severe subfertility in natural mating, a higher proportion of M2 stage oocyte with visible meiotic defect, a lower rate of zygote reaching the 2-cell, morula, and blastocyst stages, and also higher proportion of blastocysts with developmental abnormalities. So clearly, all these results imply that Patel2 play an important role in oocyte maturation in mammals. So in this curve, in fact, represents the transcriptional activity during the 3 phases, so the oocyte growth, the meiotic maturation, and the early embryogenesis just before embryonic genome activation. And what is very striking in that you can see that during meiotic maturation and embryogenesis, you have absolutely no transcriptional activity. What does it mean? It means that, in fact, the meiotic maturation and the early embryo development rely on the controlled translation of stored transcript produced during the oocyte growth and known as maternal factors. So, moreover, we know that from the Xenopus that Patel2 is an mRNA binding protein, so we wondered if Patel2 invalidation affects transcript level in mice, and if yes, which transcript. So to do that, we performed a transcriptome analysis comparing a Patel2 and a wild-type GV and M2 oocyte. So first, we measured the total RNA content from wild-type and knockout oocyte at 2 stages, at the GV stage and the M2 stage. And you can see clearly that there is no difference in mRNA quantity. However, if you look using Affymetrix microarray, the expression of all the transcript, so transcriptomic analysis, you see clearly that you have a deregulation of specific subset of genes, either downregulated in GV or M2, or upregulated in GV and in M2, and some genes are downregulated in both stages and upregulated in both stages, sorry. So we performed a gene ontology of all these transcripts, and so we were very interested because we saw that most of the genes are highly relevant in the oocyte physiology. So you can see that you have, for instance, one very important gene in oocyte maturation, which is a spermatogenesis and androgenesis helix loops helix 2, which is a transcription factors. You have also genes known to slow down oocyte maturation, which is a progesterone receptor. Also, a lot of protein, very important for physiology of oocyte physiology, controlling oxidative stress, but also DNA damage, such as glutathione transferase. Also, in regard to what we showed on the spindle, we see also defect protein involved in spindle assembly and stability, such as PAC4, CCDC69, and SKA2. And also a very important protein for the completion of meiosis 2, which is the phosphatase CDC25A, protein involved in the interaction between cumulus and oocyte, which I recall you that oocyte growth and cumulus cell, there is a strong interaction between both type of cell. And finally, also protein known to be important for implantation and embryonic growth, such as a protein belonging to the ephrine family A1, A2, A4, but also prolactin A8, A2. So all these proteins were validated through qPCR experiment. So in conclusion concerning the PATEL2 work, so we have shown that, in fact, the absence of PATEL2, an RNA-binding protein, you can modify the transcription of a subset of genes during oocyte growth, leading to a transcriptonic deregulation in the GV oocyte, and then deregulating maternal factors involved in the production of M2 oocyte, and then we have a decrease of the M2 oocyte associated with meiotic defect and blockade, such as abnormal spindle, misaligned chromosome. You have also maternal factors, very important for oocyte fertilization. So we saw a lot of defect in the zygote, such as polyspermy, partial decondensation of chromosome, or also no oocyte activation. And finally, you have also maternal factors involved in embryogenesis with a low developmental competence of embryo. So overall, so the general conclusion of my talk concerning male and female infertility, so the progress is made on severe infertility now allow us to characterize the molecular etiology, but also the molecular pathogeny. And all this result will immediately improve the diagnostic and the patient care. Moreover, it will pave for the future the road of personalized treatment in infertility. So I want to thank all my teams, but also all my collaborators coming from different countries. Thank you for your attention.